Station, this is Houston. Are you ready for the event?
Houston, this is Station. We are ready for the event.
Centers for Disease Control, this is Mission Control Houston. Please call Station for a voice check.
Station, this is Dr. Inger Damon with the CDC. How do you hear me?
Inger, this is Kate. I've got you loud and clear.
Hey, Kate, how are you? So, Kate and Shannon, it's great to meet you, and greetings from Earth. So I guess to start, to open this conversation a little bit about the three of us. And so Shannon, this is my first time meeting you, but Dr. Shannon Walker is a physicist who never lost sight of her life goal. After watching the Apollo 11 moon landing on television as a child, she started her professional career as a robotics flight controller for Johnson Space Center before being selected by NASA to be an astronaut in 2004. Dr. Kate Rubins spent a significant amount of her life in a spacesuit, whether in a BSL-4 lab suit, and now in space. Before becoming an astronaut in 2009, she was a scientist studying viral diseases and immunology, was a guest researcher here at CDC early in the 21st century, and I'm Inger Damon, Director of the Division of High Consequence Pathogens and Pathology at CDC, and it is so very 21st century to be chatting with you who are on the International Space Station. So welcome. So I guess the first question, Kate, since I know you the best, and you and I both worked in the BSL-4 labs here at CDC on aspects of the smallpox research agenda, a lot of people may not see the parallels between the work that happens at CDC and becoming an astronaut, but you made that transition from the lab bench to the space station. And can you tell us about the route that you took, which has certainly been a complex one, working in microbiology at CDC, starting your own lab at the Whitehead, joining NASA, and now here at the space station.
Hey, Inger, thanks. First of all, I want to say welcome to the International Space Station. You're up here along with the CDC, and it's wonderful to have you join us. So, yeah, it doesn't seem at first like there's a lot of parallels between lab work and being an astronaut and being in space. But, you know, we could probably grab you from the CDC and fly you tomorrow. The work that you guys do is actually pretty similar to a lot of the training that we do. So we call them spacesuits, right, when we're working in the Biosafety Level 4 laboratory. And then we actually do wear spacesuits up here. They're pretty big. They're white. The ones up here have a little bit more mass. They're about 300 pounds, but there's a lot of parallels. So working on things that are high consequence, working in situations where you have to take care of yourself and your teammates. There's consequences to your action, and also working in an environment where lives depend on you. I think everybody at CDC is really, really, really familiar with that feeling that their work has lives dependent on them.
Well, that's, I think, a great lead into, I think, current events of where we are now. And I think everybody knows the past year has been unprecedented with the COVID-19 pandemic. And so, Shannon, can you tell us about the precautions that you both had to take before launching during the middle of this pandemic?
Yeah, it got very complicated very fast when the pandemic hit and we were in the middle of training. And so first we had to figure out how to accomplish training amidst the pandemic. And so we all took a step back. We figured out what training we could do remotely and what training absolutely had to be done in person. When we did training in person, everybody, of course, was wearing masks. We kept our distance to the extent possible. But, of course, when you're training for a space flight, you and your crew have to be in the spacecraft together training. So we always wore masks when we were inside our spacecraft. Closer to flight, we all went into quarantine. In fact, our families went into quarantine with us. So we spent about 3 weeks in quarantine before we headed off to Florida in quarantine there as well. So we made sure that everybody was healthy before we launched because we do not want to bring anything up to the space station with us.
most of your time on the space station involves conducting research. And some of the work that you've done during your first mission aboard the International Space Station has involved engineering heart tissues, sequencing DNA. So, Kate, you're the first person to sequence DNA in space, and exploring the mechanisms of bone density and muscle mass loss. Can you guys tell us what kinds of research that you're working on now?
Yeah, we're continuing a lot of the biological research. So I happen to be lucky this mission to get the next flight of the cardiovascular experiment. And this was a more complex tissue mix. So we're actually looking at three-dimensional tissue engineering up here. And in addition to the cardiomyocytes, we added in fibroblasts and endothelial cells. So we're able to model the heart tissue better. We're continuing the sequencing. We're multiplexing now. We just started multiplexing about 8 different swab samples that we've taken around the space station, and I've already told them we need to get this into 96 well plates and scale this up to high throughput. So I'd like to do 100, 1,000 samples. It is more complex up here to do things like molecular biology, and so some of the time we are just inventing how to do lab work. We recently found that you can use a Sharps container as a disposal for pipette tips, and this works great on Earth. Of course, the pipette tips go to the bottom of the Sharps container. Up here, we actually have this kind of static cling that will keep them in the Sharps container instead of floating out. So sometimes we're engineering some very basic and rudimentary things. Sometimes we're studying complex particle physics with the Cold Atom Lab, which Shannon can probably explain a lot better than I could. There's an amazing amount of other kinds of research, not biological, things like combustion research, fluid behavior up here is just completely changed. So there's about 200 experiments on any given mission, and it's pretty fun to be able to do them all.
Well, I imagine that fluid behavior must make sequencing a real challenge, especially when you're getting to 96 well plates. So are there new aspects of research that you're embarking on because of the COVID-19 pandemic? I know you mentioned some of the higher throughput sequencing that you're thinking about doing.
Yeah, so we're not specifically studying any pathogens of consequence up here above BSL-2. That's all that we're rated for. We have had some impacts because of COVID. For example, in our research teams on the ground, like everybody else, they've had to move to mostly remote work, and then when they have to be in person, doing masks and distancing. And so there were some delays for a while, but it's actually been incredible to watch the teams at NASA still be on a launch schedule. So it wasn't just the Soyuz launch. They then launched Crew 1 was the first operational flight of the SpaceX vehicle. And we've had cargo vehicles come up with literally tons of supplies and experiments, and those are packed and manifested by ground teams that are having to figure out how to do all this. So there's been some impacts to research in terms of operations, but overall, everybody's really done an incredible job of keeping this space station supplied and running.
So how long will you both be on the space station this time?
Both of us are up here for about a 6-month stint. We actually, I guess Kate knows her landing date. I'm not quite sure about mine. She's been up here about a month longer than my crew has been from the SpaceX Dragon vehicle, so I figure 6 months-ish.
So I think, here on terra firma, I think and I think with COVID, we certainly are beginning to see new things about the SARS coronavirus 2 pathogen. But I think, for the most part, as we begin to understand pathogens, they behave in a typical way. What have you been learning up there about how microbes act in space? And is there information that you're beginning to get from some of the sequencing?
Yeah, that's a great question, and actually it's one of the experiments that I've been working on this week. So we're working on a comprehensive map, sequence-based map of the space station. We're taking about 1,000 samples total, and so I'm very carefully swabbing all of the modules. Everybody floats by and says, "Kate, are you still swabbing?" And I'm like, "Yep." 1,000 samples is a lot. We're really trying. We've done some early microbiome studies where we do a smaller number of samples, but with this one, we're interested in a really comprehensive three-dimensional map. We do know some information about the way that microbes change in space, and there's not just the behavior of the microbes in terms of their gene expression, what products they're producing, but also who's up here. So if you think of the microbial community as a census, we're trying to take a census of which microbes are on space station, which maybe we carry up here and find it a favorable environment, which we don't even bring up here because we don't have a lot of things like soil coming up here. So we want to know who's up here, and we want to know how they're changing. In terms of viruses, we actually have done very few studies. We do know that astronauts are a little bit immunosuppressed, and we can get things like VZV reactivations. This is a virus that causes chickenpox and shingles later in adults. We've seen that before in astronauts. But we don't have a lot of information about basic virology up here, and I think this would be pretty fascinating to study.
Well, I'm sure there's a lot of folks at CDC who'd be interested in trying to go up and help out with some of that work. But so I guess to go back to bacteria, so my division at CDC works on a number of rare bacteria, one of which is called Elizabethingia, and it's named after the CDC scientist who first identified it, whose name was Elizabeth King. Folks may be interested to know that one species of Elizabethingia was first identified in space, actually, in the condensation water on the space station Mir, which Russia had in orbit from 1986 and 2001. Does it surprise you? I mean, again, you sort of alluded to this in thinking about the microbiome and what it looks like there and how it's influenced by who's on the space station, but does it surprise you that we might find the same or different bacteria in space?
Yeah, I think it's incredibly interesting to see what it's like in a completely artificial environment. And so you can probably see a little bit in the background here. We're surrounded by four walls of pretty technological things. So we don't have, for example, houses that we're going to. We have no dirt that we're interacting with. We don't have anything that you would see in the natural world. And so this is a completely man-made artificial environment. And so it's pretty interesting to try to understand which pathogens would survive up here, which surfaces they would colonize, who would grow well. For the most part, we don't have a lot of pathogenic bacteria up here. We do culture-based methods for a long time, and we're really starting to get into the sequencing now. But it is interesting to understand how microbes might adapt to this new environmental niche in space. And I think a lot of the sequence-based technology will uncover things that we just haven't picked up. We've been here for 20 years on the International Space Station, but culture-based methods may not be picking up kinds of interesting things that we see up here. And so doing these comprehensive surveys is probably going to discover a lot of new things. If you have folks that need to isolate and grow up some cultures, we sure could use the help.
So there's been now some promising research collaborations between CDC and NASA, and the technologies that you guys are working on are helping to enhance data on environmental hazards that we're beginning to accumulate. And there's real interest in the sampling of the interior of the living spaces for antimicrobial resistant organisms. What other sorts of collaborations between CDC and NASA do you think can benefit from both of our current work, other than culturing viruses in space?
Yeah, so a lot of what CDC does is really a huge strength is the epidemiology work, and it'll interest you to know that we actually do quite a bit of that at NASA. And so we've got a very particular subset. We're looking at astronaut health, and we're looking at long-term effects of space travel. But I think that the expertise at CDC with the epidemiology aspect and this very kind of unique cohort could definitely benefit from a collaboration. And then I think also things like the wet lab work in terms of understanding how to process samples, how to identify samples, how we could increase the efficiency and the throughput on the International Space Station. I know at CDC you guys process thousands, hundreds of thousands, millions of samples, and so you understand how to do that kind of work, and we understand how to adapt that to the space environment. And then also computationally, so we're very interested here in trying to figure out eventually when we go to Mars, and then also for the lunar surface, how are we going to be identifying organisms? Does that database reside on Earth? Does that reside up here? Understanding the flow of information and how we can take a look at these things computationally, and that's also really a CDC area of expertise that is done very well.
Yeah, and a big area of recent focus as well, just thinking about that computational work. So Shannon, you look like you have something to add. No, okay. So I guess I'm going to move on to some questions submitted from people across the agency, so our rapid fire questions. So the first one is, can you see the Milky Way from space?
Absolutely, you can, and it is even more spectacular in space since you don't have an atmosphere to get in the way. The number of stars you can see is stunning.
Oh, amazing. So question to both of you. Who inspired each of you to go to space?
Well, I think you already told my story. It was the early moon landing that inspired me.
I'm not sure where I got the inspiration to go to space. I just always wanted to be an astronaut. My parents definitely encouraged that, and they let me go to space camp, but I did have to work for it. So I had to mow the lawn, and I earned $3 an hour. So I'm going to credit them with a lot of inspiration for hard work, and it's sometimes good when you have to put some skin in the game for your dreams.
So, well, when growing up, I had to clean the bathrooms. So, and I think I got about $3 an hour. So the next question, what was the first thing that you ate in space? And Shannon, I understand that you like to cook. So have you experimented with cooking in space? Any success stories to share?
Yeah, we do have some experimenting that goes on up here. I think at Thanksgiving, we didn't get our expected food supplies for Thanksgiving, so we had to improvise. But we did have some what is it? Some cornbread dressing. We had a can of crab meat, and we had some sauce that I made, or that we had. So I sort of put together some deconstructed crab cakes for our Thanksgiving dinner. So that was fun. And then the other day, we actually got to harvest some of the plants that we're growing, and so we made some improvised salads that were pretty good.
So what's the condition of how loud is it in the working environment aboard the space station?
Yeah, acoustically, it's actually it's an incredibly loud environment. We have a lot of folks that monitor our hearing protection up here, and we actually evaluate each payload for what it's contributing to the acoustic spectrum. And I think it's about 50 decibels in our crew quarters, which is the quietest place on space station. It can get up to like 85 when somebody's running on the treadmill. We limit that noise protection. We do have hearing protection up here, but it's actually I think that's a really interesting area of research, because we're surrounded by pumps and fans. So we live in the middle of our life support equipment. So those kind of maintenance floors on all of your buildings, that's where we sleep and we eat and we do our work here. And so monitoring that acoustic level, understanding if it's a hazard to human health, it's a really big deal in space because we're in this environment for so long.
So do you wear like the little earplugs like what we wore in the BSL-4 lab?
We do wear those to sleep if we can, but then on the other hand, we also have to be able to hear any alarms if we get a caution or a warning alarm that goes off in the middle of the night that signals a space station problem. So you want to block the noise, but you also want to be able to hear the speakers and the caution and alarm. So if CDC's got any interesting technology for acoustic monitoring and or noise prevention in the background, we'll take that too.
Okay. Yeah, maybe you need some more strobe lights like we have in the BSL-4 lab. But so I think we are being asked to wrap this question. So I'm going to ask you one more question, which is from folks in the agency. The world has been dealing with social distancing measures over the past year due to COVID-19, and certainly you have an interesting perspective on that being on the space station. And what has the experience living on the space station taught you about maintaining mental health under isolating environments?
Yeah, we actually get that question a lot, and I know some of our crewmates have given talks to address this. I mean, the important thing is to try and stay connected somehow to your friends and family, and we do that similarly to the way people are doing it on the ground. We have phone calls that we can make from the station down to our family, and we are able to do video conferences every once in a while. So that's how we contact the people on the ground, and then we realize that the people we're with, of course, are our space family. So we get to spend time together, which is really nice.
So hopefully you don't have too many arguments with your space family. But no, we really appreciate your spending the time talking to us. I see some head shaking there. And it's been just a great pleasure to have the opportunity to talk with you guys on the space station. So thanks so much for the opportunity. Good luck with everything that you're doing, and we really appreciate the time you spent with us.
Thank you, Inger. Thank you so much to all the Centers for Disease Control. You guys are doing an incredible job to keep our United States safe. And from the International Space Station, we really appreciate it. Thank you.
Station, this is Houston ACR. That concludes the event. Thank you.
Thank you to all participants from the CDC. Station, we are now resuming operational audio communications.